Hello, everyone, and welcome to today's webinar, AI Microscopy Symposium, 7th edition. I am Kaylee Bach of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Leica Microsystems. To learn more, visit www.leica-microsystems.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I will now pass it over to Luciano Lucas to introduce our speakers.
Thank you very much for the introduction. It's really a pleasure to be here today. My name is Luciano Lucas. I'm the Director for Data and Analysis here at Leica Microsystems, and we have a wonderful lineup today of speakers. This is a topic that is close to my heart, of course, and close to the heart of Leica and the team that is represented here. The AI Microscopy Symposium is something that started already some years ago, in 2020, and thank you for all of you that have participated in the previous 6 sessions, and also thank you and welcome for attending today's session. Wherever you're joining, good afternoon, good morning, or good evening, as it may be the case for some of the speakers we have today are actually in Australia, so it's very late for them. If you're interested in looking at some of the presentations that happened in the past, they are on the website, on the Avia website. You can see them right here. Okay, here. Yeah, here they are. And there's quite a lineup of speakers that attended, participated, and presented their work. Either exciting data set, exciting tool, or exciting new algorithm for machine or deep learning. And we have all of them present there, and you can navigate and watch any of those. Today, we have 4 speakers, but 3 presentations. First, from the University of Melbourne, we'll have Vijay Rajagopal and Afshin Khadangi, and they will be presenting their latest work that is, of course, very, very exciting. And it's the second time already they're presenting here. Afshin is currently at the University of Melbourne, working towards his PhD, still in Vijay's laboratory, and they will be talking about their latest work. Some of it will include the cell physiome, which aims to predict the single-cell biology. They also have worked recently on the EMNet, which is a deep learning for electron microscopy image segmentation. And I think what is going to come up today a lot is the Cardia Engine, which shows building blocks for virtual cardiac cell using deep learning. So this is all very exciting. This comes out of Vijay's lab, and Afshin is a graduate research assistant currently. Vijay himself is at the University of Auckland, Melbourne, my mistake. I'm looking at my wrong notes here. And he is the principal investigator for that presentation. The second speaker will be Yicong Wu. He's currently based at the NIH in Bethesda. He is there a staff scientist already for a number of years, more than 10 years, and he gained his PhD at the Hong Kong University of Science and Technology. He works with Hari Sharoff and a number of other top scientists at the NIH, and they focus on using deep learning to increase resolution or increase the quality of the images. So I'm looking very much forward to this presentation as well. And the final presentation today will be from Parmida Ghahremani, and she will be presenting deep leaf, I expect, and crowd deep. Those two presentations or those two topics are very important. This is about multiplexing, looking at how to use deep learning for the analysis of images and then extracting insights from spatial data sets. So very exciting lineup of speakers today. And with that, we should get started with Vijay. Over to you.
Hi, thank you, Luciano, for your kind introduction, and thank you all for coming to this symposium. It's a really exciting symposium, and I do want to thank our organizers here for inviting Afshin and me to present our research, our most recent research. So let me get started. What I'm going to be talking about today, what Afshin and I are going to talk about today, is a new deep learning pipeline called Cardia Vinci, which enables us to create computer models of the cardiomyocyte using generative adversarial networks. Before I get into, or actually before I let Afshin get into the nitty-gritty details of it, I just wanted to give some context to what we're going to present today. In the way, by introducing you to the type of work that we do in our lab. So as Luciano indicated, we're at the University of Melbourne. I'm in the Department of Biomedical Engineering, and our research group is really have expertise in creating structurally and physiologically realistic models, computer models of organs and cell systems. And one of our research aims is, or research goals, is to develop a digital twin model of a living cardiac muscle cell. What we mean by that is that we want to construct a detailed structural model of the ultrastructure of the cardiac myocyte using the latest electron microscopy and super resolution microscopy kind of imaging techniques. We want to, and within those geometric models, we want to define mathematical equations and computational simulations of calcium dynamics, which drive every heartbeat. We also are working towards building more detailed simulations of the energy requirements in the cardiac muscle cell during each heartbeat, and indeed during development and growth and disease. And of course, the heart muscle's primary function is to beat, and so we're also defining mathematical equations to describe the mechanical contraction of the cell from different types of biological data that are now sort of exploding into our research community, thanks to all the latest technologies. And so the thing that we want to do by implementing these different mathematical models in these structurally detailed geometric models is that we want to take those models and then generate insights on disease mechanisms that the heart sort of undergoes in conditions like diabetes, heart failure, so on and so forth. And when we work with these different types of data, invariably we have to deal with different types of structural data, and electron microscopy is one of them, because we get a lot of really high resolution detail of the ultrastructural changes that occur inside the cardiac myocyte using this technology. And what Afshin is going to present to you today is the innovation that he's been making in helping us make this process of incorporating structural detail into our models more efficient. So with that, I'm going to now hand it over to Afshin to present to you what Cardia Vinci is about. Afshin?
Thank you, Vijay. Greetings, everyone. I also wanted to thank Luciano for organizing and his team for organizing this amazing symposium. And I'm just going to walk through the Cardia Vinci, the methods that we have used, and the idea, the main idea behind the Cardia Vinci that drive us to actually obtain a virtual 3D reconstruction of cardiomyocytes. So the initial driver for studying the Cardia Vinci and the research that we went through was basically that we all know that these days with the advances that we have and we see in electron microscopy, we can actually obtain unprecedented and large three-dimensional views of cardiac structures or other tissues. But one of the main challenges during analysis of such data sets is basically that it takes much time to extract, segment, or even statistically quantify or analyze the 3D changes in these tissue types. As Vijay said, we want to just expedite this process because we could have different conditions, different disease conditions, and given that variety of the conditions that a cardiac cell could go through, we want to expedite the statistical analysis and just get more rapid results in the 3D changes that we can see in the cardiac structures. So, having said that, with all of these advances and also the limits that we face in the 3D statistical analysis and quantification, we studied and proposed Cardia Vinci as a platform and framework that uses deep neural networks to automatically segment and also statistically quantify the morphologies that we observe in the cardiac cells. So what is the workflow for Cardia Vinci? So basically, as you can see on the right figure, it initially starts with the segmentation of mitochondria, myofibrils, and Z-discs. So we are using semantic segmentation as the first step, where we segment mitochondria, myofibrils, and Z-discs together. And once we have segmented these ultrastructures, the next step is actually feeding those segmented image slices to train a modified version of the style GAN. And I will talk about why we have used style GAN in the methods section. But so once we have actually trained the style GAN, we are able to project the latent space to obtain the projections and generated images using the trained style GAN. And once we have generated these images or these sliced images from the style GAN, we are using Jacob distance to sort the image slices by minimizing mutual Jacob distance between the generated slices. So once the process of minimizing of Jacob distance has been done, we rearrange the GAN generated image slices accordingly to obtain a 3D image stack and reconstruction of the cardiomyocytes that are actually projections of the latent space. And once it had been done, we can obtain 3D shape and geometrical statistics. So we can quantify the changes in such statistics that are vital to study 3D changes in the presence of the disease or other conditions. So I'm going to just briefly talk about the methodology that we have used in our study. So for the data sets, we have used 2 data sets for our study. The first data set was basically a publicly available data set, which is a focused ion beam scanning electron microscopy data set for the controlled cardiac cells. And the second data set was actually involving a serial block-based SVM dataset of the left ventricular cardiomyocyte, which was actually extracted from type 1 diabetic rat tissue sample. So once we collected the dataset and the curated dataset, manually labeling, etc., we trained and tested our neural networks. So all of these training and testing had been done on AWS and partially on high performance computing facility called Spartan at the University of Melbourne. And then the semantic segmentation. For the semantic segmentation, we used a modified unit with where actually we had modified the bottleneck as a densely connected convolutional layers, and also as activation functions we have used trainable linear units. And we also finally modified the SciGAN by introducing an auxiliary loss function to minimize the distance between one hot encoded image formatting that we had passed along the TF record dataset to the network. So in terms of the results were interesting because one of the results that actually caught our really attention, and that was one of the main, I would say, surprises in this study, was basically the accuracy and the segmentation performance metrics for the Z-disk segmentation. Because most of you might know that basically Z-disks are really thin components of the cardiomyocytes, and they are really challenging to trace and segment. And we had conveyed previously a research around segmenting Z-disks solely. And we showed a couple of results there that it is really challenging to segment such components, and mainly it is due to data imbalance, because it is just comprising less than 1% of your dataset in your image dataset. And but when we use semantic segmentation, we noticed that actually Z-disk segmentation shows really promising and high performance metrics compared to mitochondrial Z-disk, as shown to you in a couple of important metrics, including variational information score, and also a couple of other metrics as shown to you in this figure. The second result is definitely that we could actually generate realistic models of cardiomyocyte architecture. So the architecture that you're looking at actually is the generated and is the reconstructed volume using using generated image slices that have been projected from the latent space. So we have projected the latent space slices and then reconstructed according to an algorithm and according to a logic, which is centered around minimizing jacket distance between these slices mutually. And then we were able to reconstruct this volume. But on top of the the visuals that we can see in those kind of figures, we use a couple of tests and also a couple of metrics to validate our results. So we use ANOVA testing, and as shown to you on the left table, we have validated a couple of metrics, geometrical metrics, and also geometrical statistics, and also shape statistics in 3D. And we have compared the results of what we have obtained from GANs and also the real sample. So we have compared these two. Also on the right image, you can see that we have compared the mitochondrial myofibril relative density frequency by sampling a volume of 0.4 microns in each axis. And once we have obtained that sort of volume, we have just calculated the density of the mitochondria to myofibril volume and obtain finally a frequency distribution. And you can see that it is exactly fitting the same as the actual data set. So they are similar in a sense. Okay, so in this video, we have actually rendered a 3D version of the generated cardiomyocytes. So blue is Z-discs, green is mitochondria, and red is myofibrils. And we have also rendered a limited, I would say, a couple of slices from the same volume to get a closer look into how these are actually, how these components and alter structures are actually comprising this volume that we are looking at. So the volume that you're looking at the moment is actually generated as the GAN output. So we have reconstructed this volume from the GAN generated slices. And again, blue is Z-discs, myofibrils are in red, and mitochondria in green. And I presume that you all noticed that myofibrils and Z-discs are actually aligned in planes exactly similar, in the similar form and shape. And so finally, so as a result of CardioVinci, once we went through the CardioVinci and proposed the CardioVinci, we were thinking about integrating all of our findings and all the methods that we had proposed so far into a simple and unique project called Cardia Engine. So the aim of the Cardia Engine, as you can see here in this image, is basically taking raw images, raw EM images of the cardiomyocytes, and then generating a geometric model. And once we have obtained geometric model through channels like Cardia Engine, let's say, then we want to obtain some computational insights from the generated volume, as Vijay highlighted before. So that's the main idea behind Cardia Engine, and I would say that we are looking to actually integrate all of our findings into this unique project. And finally, and more importantly, I want to thank our colleagues and our collaborators. So I want to thank Professor Bodia, Professor Hansen, and Dr. Brian Glancy for providing the data sets for this study. And also, I wanted to thank April for her assistance during the annotation of our data sets. And huge thank to definitely for Spartan facility at the University of Melbourne for providing freely accessible high-performance computing to the community. So that's from us, and feel free to follow our updates on Twitter. Thank you.
Very good. Thank you very much, Ashvin. That was a very good update on the progress that you're making. There are a couple of questions that came in the meantime. One of them comes from Wang Yang Choi, and the question, well, I'll read out what she wrote. Thank you for the great talk. Regarding cardio Vinci, what's different about cardiac tissues that requires this deep learning workflow? And she has some suggestions. Elongated muscles, can this workflow be modified for other types of morphological detection?
Thanks for your question. That's a great question, definitely. According to our experience within the last couple of years, we had another study on top of EMNet, which is called EM Stellar. In EM Stellar, we showed that these neural networks are sometimes tissue agnostic, and sometimes they are not. So some of them are actually showing really solid and good performance metrics for segmenting, for example, mitochondria only, but they fail. I'm talking about just one tissue. I'm not talking about across different tissues. Even in one single tissue, segmenting a couple of components could be challenging compared to the others, as we have shown many, many times. But for your question, I would say that definitely the cardiac structure always is amazing to me, because you can see the sort of repeated components across your data set, and that makes it relatively I would say easier compared to neuronal segmentation, according to our experience. But I would say, depending on the ultrastructure that you are trying to segment or extract the architecture for, or even depending on the dataset itself. For example, serial block phase segmentation could be different to focused ion beam dataset, and depending on the resolution. But anyway, if I want to put all these together, I would say definitely the workflow. I'm not sure what do you mean by modified, because the workflow we have already tested for diabetic and control datasets, and we have shown that it actually works across 2 different datasets which have pretty much different resolutions. We are talking about 20 nanometers and 15 nanometers. So that's a huge difference. But in terms of the tissue type, we haven't actually tested the Cardia Vinci for other tissue types. So I'm not 100% confident that I can say you can absolutely apply to different tissue types. But that's what we have highlighted in our paper, and we have encouraged the community to use Cardia Vinci and apply to different tissue types, and hopefully they can get actually the results that are statistically significant, as we have shown in our study. So I hope I have answered your question.
Thank you. There is no follow-up question yet, so I do have one question too for you and the
Please.
team, right? I'm curious about your experience using GANs. You said that initially you were using UNets. What was the role of using the degenerative adversarial networks for the project here?
Yes,
What was the
absolutely. Yeah, 100%. So you can imagine that to statistically quantify the data sets, you need to collect a large number of tissue samples. And having said that, once we collect all these sort of data sets, we need to also sit and spend time, sometimes for annotation, not essentially, because sometimes you can use some sort of transfer learning or tweaking your CRAN network to segment other tissue as well. But anyway, if you have collected that sort of, let's say, tissue types, you need to spend time around training, testing and deploying, and finally segmenting all the structure and then quantifying in 3D. So the main driver for using GANs here was basically, is there any way that I can actually quantify the distribution that the data has been generated from? And if I can actually, so that's the main idea behind the GANs, right? So GANs are actually assisting us in getting the distribution of the data set of the current data set that we are dealing with. That data set could be generated from that distribution. And once we have obtained that sort of distribution, we can sample from that distribution and generate different versions of the data set that actually share the same statistical properties with the current data set that we are dealing with. So the main driver for GAN here was basically to obtain some fakely generated cardiomyocyte images that share exactly the same properties as the actual dataset or real dataset that we are dealing with. In other words, we are mocking an algorithm or deep neural network, mocking in a way that they are actually able to generate realistically looking cardiomyocytes, but different, I would say, angle, and we have already quantified and statistically shown that the results produced in 3D our results are actually showing that they actually represent and exhibit the same statistics measure in terms of the 3D geometrical statistics and also 3D shape statistics.
And this opens up, and we need to move to the next speaker, but this opens up the possibility, if you can generate realistic looking cardiomyocytes in a somewhat deterministic way, it makes it possible for you to train segmentation models also in a deterministic point, because you can have the segmentation and the generated cardiomyocytes as training pairs for a segmentation model.
Absolutely.
So that's very clever approach to the segmentation problem. So thank you so much for the presentation.
No worries, pleasure. Thank you.
Both you, Vijay, and the whole team, congratulations on this several steps forward since we last heard from you. We will go to the next speaker. Yicong Wu is our next speaker, and then we'll be back for Q&A on his presentation as well.
Hello everyone, I'm Yicong Wu from NIH. Thanks for coming to this symposium. And today I will share with you our research on the improvement of three-dimensional resolution in fluorescence microscope with multiple deep learning methods. In this talk, I will begin with the background on image restoration methods in fluorescence microscope, including classic Richardson-Lucy deconvolution and purely data-driven deep learning networks. Then I will talk about our recently developed methods, Richardson-Lucy network, RLN, that incorporates the image formation process into deep learning. Last, I will show you their biological applications, including CARES, RCAN, DDN, and RLN to improve fluorescence microscope performance. In live fluorescence imaging, there is always a so-called pyramid of fluctuation. With a limited photon budget, you need to trade off between the spatial resolution, temporal resolution, signal-to-noise ratio, and how long you can image the live samples before photo bleaching and photo damage dominates. An image restoration with computation methods can enlarge this pyramid. Microscope image usually looks blurry as the true object O is blurred or convolved with the point spread function or forward projectors of the microscope F, and then added with noise, either Poisson or Gaussian noise. And Richardson-Lucy deconvolution methods has been widely used to explore the recovery of microscope images. And this is an iterative process, and here shows the equation. And if you have an initial estimate A, you first convolve with the point spread function, and then compare with the measured image. And then you convolve this division with the flip version of this forward projector. And then you update your estimate, get a new one, and you iterative this process until you get converged. And usually this requires tens of iterations or even 1,000 iterations to get fully converged this EK to the true object. And recently, we developed a Richardson-Lucy deconvolution method with unmatched back projectors to speed up this process. The idea is to use a new term, back projector B, to replace this flip version of the forward projector. And we found out that if the product or if the convolution term between the backward projector and the forward projectors is close to a delta function, or their frequency domain, the multiplication close to 1, then we can achieve the full convergence with one iteration. But both Richardson-Lucy deconvolution methods and unmatched back projectors has some challenges. The first one is computation testing, and regular one you need to large number of iterations. In some cases, to deal with those noise data, you want to introduce some regularization term, and this will cause extra computation. And even with our new variants, unmatched back projectors, we need to carefully design the parameters depends on the different microscope, different numerical aperture, and different image quality. In both methods, the stopping criteria remains a problem, and this stopping criteria is quite subjective. Usually you just manually or visually check the image quality at different iteration steps. The purely data-driven neural network has also widely explored to restore the microscope images, and these methods includes content-aware image restoration networks, CARE, which use a UNet, and residue channel attention networks, RCAN, that we have collaborated with Leica and S-Vision to develop 3D RCAN to denoise to improve the resolution at and beyond the diffraction limit. We also develop a dense decon net, DDN, based on deconvolution, a dense net, which can also deblur images, especially for those blurred with spatially variant PSF. All these neural networks has some challenges, and we feel that interoperability is a problem because it's just a black box. You don't know how the train is in the intermediate step, and generalization ability is another problem. You want to apply a model to another different data set or different structures, it might be difficult. So recently we developed a Richard Sanusi network, RLN, that combines the forward and backward projectors in the convolution term and the fully convolution structures of deep learning networks. And here is RLN and RLD. Let's go through the RLD again. We can split this into 4 steps: the forward projection step, the division part, then the backward projection, and then we update. And in the RLN, we also have these 4 steps, and this RLN is consisting of 3 parts, H1, H2, and then merge this H1 and H2. So in H1, we downsample the input data by a factor of 2, and then run the forward projectors with multiple kernels, but small kernels. And then we do the backward division, backward projections, upsample back, and then update the estimates, and then we get a new one, E1. And for in the H2, we use the original data, but use this kernel with these layers and do the same, these 4 steps. Then we get the E2. Then we combine the E1 and E2 as in the third H3. And with this design, H1 and H2 can provide complementary information so that we can maximize the use of GPU. And here I want to show you the RLN's interpretability under conventional test. That means the training data set and test data set has the same structure. This example is a GLP labeled cell membranes in a C. elegans embryo acquired with light sheet microscope. And here compares the lighter view and ICL views, and ILD FP1 is the first iteration, and FP1 in ILN is in the H1 block, and this is backward projectors in ILN, and then ILD F5 iterations, and F forward projectors ILD in the H2 block, and backward projectors. And here is ILD output and ILN predictions. And you can see that these intermediate steps at ILN is very similar to the ILD. This means that we will incorporate these ILD's intermediate steps into the network, and that makes this network becomes interpretable. And this shows the generalization test, and the test data is a human brain phantom, and the training data is different than this data set. It's just a synthetic phantom object consisting of different structures like dots, spheres, and some hollow structures, and both data sets are blurred with the same PSF. And again, we compare the lighter view and ICL views at different intermediate steps. And you can see that these intermediate steps are very close between ILD and ILN, and then ILN prediction results actually outperform the ILD. I will show you in the next few slides. But here we compare the learning parameters and processing time of ILN versus other purely data-driven networks, CARE, RCAN, and DDN. The learning parameters between RN and DDN is close, but it's much less than CARE and RCAN. It's only 1/60. And for the training time, it's close to DDN and CARE, but it's 3 times faster than RCAM. And for the prediction time, RLN is 4 times faster than CARE and give us 50 times faster than RCAM. So here we compare the image restoration performance between these different networks. And here is fluorescence beads under the conventional test and lateral view and ICO views. You can see that CARE, RCAM, and DDN, no matter in the lateral view or ICO views, you can always see some distorted recovery. But RLN is better close to the ground truth. And here is the phantom tissue under the generalization test. And here shows the zoom in region of these ICO views. And you can see that only RLN can well predict these sharp structures, and ILD, CARE, RCAM, or DDN all fails to predict these dots. And we quantitatively analyze the SSIM and PSNR values and found that in both this case of this brain sample case, ILN give us the highest SSIM values and PSNR values. And now I will show you some applications of this RLN. And here is on the wide field data with cells, and this is generalization test. And the training model was based on the synthetic phantoms, not the real structures of these cells. And the red color is mitochondria, and green is actin, blue is tubules, and yellow is the nucleus. And here is the raw wide field, and this is the direct Richardson Lucy results. And this is RLN predictions, and this panel shows the zoom in region and in lateral view and also in ICO view. You can see that RLN prediction can better resolve these fine structures of these filaments can better separate, and because these autofocus cannot well be deconvolved with this ILD, but RLN can better shows this recover these results. And this example is the same with wide field microscope, but it's a thicker sample. It's a C. elegans embryos expressing TTS3B GFP. They label the different neurons. And when you compare the raw data and the RLN output, you can see that RLN prediction can well separate these neuroids. And for single neurons, RLN also outperforms ILD. These fine structures of the neuron can be well recovered than ILD. And you see this gut region. You also see that this line intensity profiles over these membranes. You can better see that RLN can separate these neuroids. Otherwise, raw is too blurry to see. And also you can see that this enhancement occurs not only in lateral view, but also in ICO view. Now I will show you other applications of these RLNs. And here is a dual view light sheet microscope, light beam. We have developed this system quite a few years ago. And the idea is if we introduce the light sheet from left-hand optics, then we collect the signal from right-hand camera. This way you get good data resolution, but the ICO resolution is poor. And we alternate the excitation from right-hand optics, then this poor ICO resolution becomes good lateral view from these left-hand objectives. So if we can merge these two views into one, then we can get isotropic resolution. And our algorithm is using joint deconvolution. So again, if we have a single view, we have an estimate, and then we use this estimate to go to the second iteration, compare with the second views, measure the image, and then we get an update. We alternate this process, and eventually we can get isotropic resolution at 330 nanometer at all 3 dimensions. So this one compares the neuron developments in C. elegans between a single view light sheet or dual view light sheet in lateral view and ICO view. You can see that the fine structures of this neuron were better resolved with this dual view light beam. And this dual view needs to register this to get this isotropic resolution. We need to register the dual views and then join deconvolve. So we are thinking if ion can predict these dual view images from a single view input, and here compares the care, RCAN, and DDN on a U2O cell transferred with TOM20, that's mitochondria. And you can see we compare the raw ground truth, that's the dual view deconvolution ground truth, ION, RCAN, DDN, and care. And you can see that only ION can better resolve these fine structures, while the DDN or RCAN care fails or cannot greatly improve the ICO resolution. And here shows the time lapse of ION compared with the raw data. And just with a single view input, we can predict this nice resolution in lateral and axial resolution. We have also used DICE beam to image large clear tissue, and here is an example of mouse brain tissue labeled with some neurons and cleared with iDisco. And our multi-view fusion can enable isotropic submicron resolution on this large data set. And because the data is so big, so we cannot directly register through data set that's almost terabytes data. So our processing pipeline is to run a coarse registration and then crop the big data into maybe 1,000 subvolumes, and then run fine registrations, joint deconvolutions, and then stitching back. Even we use the back projector, unmatched back projectors, we can still need 3.4 hours to process this data set. So we are thinking, how can we use ION to speed up this processing? So here we show that the ION prediction on these neurons, and again we compare with the RLN, CARE, RCAN, and DDN, and you see that in the later views the prediction are very close. But in ICO views, CARE will mix some structures, RN can bring some extra noise, only RN can get best similar to the ground truth. And again, if we calculate the PSNI, SSM, and only the IN can give us the highest value of these, also including the mitochondrial data. So here we test on an even large clear mouse brain tissue, and it's all about neurites. And these color indicate code different depths, and these code different lateral positions. And if we zoom in a small region and comparing the raw in lateral view and ICO views, and you can see that IM prediction is quite close to the ground truth, not only in the these fine structures, but also in the frequency domain. And compared to our previous processing pipeline, as I mentioned, the registration, crop, fine registration, joint deconvolution, and stitching, we found out this RN can improve 4 to 6 times and faster than this pre-processing method. And most important, joint deconvolution need to do the registration of the two views. So in some cases, in some small subvolumes, if joint deconvolution fails due to the error registration or some hardware failure, then we lose those details. But ILN just rely on single view input, so we can well preserve the structures and recover the resolution in these views. So here shows the entire 3D volume rendering, and this is the raw data. And then the IM prediction, you can see it becomes sharper. And then if we compare with the joint deconvolution, you see that in some cases they are very close, because if they good registration, but if not, it fails. You see the structures are missing in the joint deconvolution, but still will predict with the ILN. So if the volumes are contaminated by severe noise, so ILNs cannot fully recover the resolution. Although direct Richard-Lucey deconvolution totally failed, ILN can predict, but not as good as the ground truth. And if we just use RCAN to denoise the data, it's also not as good as the ground truth. So we adapt a two-step deep learning, first using RCAN to denoise, then use ILN to deblur, And then we find out this two-step deep learning can best recover these ER structures, and this is confirmed with SSIM and PSNR calculations. So in the following slides, I will show you more applications about these multiple-step deep learning approaches on other microscope. So the first example is multi-view non-confocal microscope. Confocal microscope is a very powerful imaging tool to image scattering samples, stick samples, but the resolution in iso dimension is not as good as the lateral dimension. And the second one is a point illumination and point scanning makes the 3D imaging very slow. So we develop a non-confocal microscope and also with two extra views. So this example is tubules on a fixed cells. So when you see from the top views, it's quite blurry along this dimension. From right hand objective, it's blurry along another dimension. And in C view, it's along this Y dimension or this Z dimension. And again, we use joint deconvolution method to combine these three views into a single one. And then you can see that the resolution after this triple view deconvolution is greatly enhanced. And here shows the triple view deconvolution results, and the color indicates different depths. And here is in the XZ view. And here I show you some live applications of this live confocal imaging. And this one is metal tracker green and the lyso tracker deep red. And comparing the raw and the triple view deconvolution in lateral and axial views, you can see that the triple view combination can greatly improve the resolution. And this is the cardiomyocyte labeled with metal tracker. And the same thing, you can see that triple view improved the views. But the challenge with this triple view non-confocal is the speed, because you need to acquire three views. And these three views will also cause extra photo bleaching and photo damage. So again, we try to use deep learning to solve this problem. And we use a two-step deep learning methods. And the idea is for each view, we denoise first, we create the denoising models. And then based on these denoised three views, we joint deconvolve them to run a triple view deconvolution to create a ground truth. Then we train another model from this denoised view, see bottom view, to this three views ground truth to get the convolution model. And in application, we only need to one bottom views denoise and then apply to this deconvolved model. We can also just train one step model just directly from this low SNR view C to this high triple view deconvolution. And here compares the results on a GLP histone in C. elegans embryos from a raw view, just one-step deep learning, two-step deep learning, and the ground truth triple view. And you see that only the two-step deep learning can best resolve and recover these structures, and one step also shows some artifacts. And this is confirmed with the SSIM and PSNR values. With two-step deep learning, we can get a SSNIM value of 0.9, but with SSIM it's only 0.7. So these two-step deep learning methods has allowed us to do live confocal imagings on the C. elegans embryos to monitor the nuclei development in the entire 450 minutes, which otherwise is impossible with the triple view non-confocal microscope. And here we compare the raw and two-step deep learning as 3D projections, and here's the maximum intensity at different time points. And you can see that the two-step deep learning can recover many missing signals with a single view acquisition. And this training and prediction is based on CARE. And here's another example so that this two-step deep learning enables light confocal imaging and also outperforms the dice beam. Again, we monitor the development of these AIB interneurons in the nerve ring, the brain in the C. elegans embryos, and comparing the raw two-step deep learning lateral and axial views And you see that these fine structures are resolved and recovered with deep learning prediction. And this two-step deep learning outperforms the dice beam at similar time stage. And this allows us to monitor the growth dynamics at the tip of neurites over the entire embryogenesis. So in the triple view confocal microscope, we can improve the resolution by structured illumination because we use line scanning. And if we modulate the line scanning intensity along the scanning direction, then with some photo reassignment method, we can get super resolution along the scanning direction. So in this case, we improve the resolution along this dimension for the view B along X prime direction in the view A, and view C along this Y dimension. And these three dimensions provide complementary information so that we can get good resolution in all three dimensions. And here is an example of on expanded worm embryos in lateral and axial views. You can see these neurite and neuron structures. And when you compare the super resolution mode to the diffraction limit mode, you can see that resolution is greatly improved compared to this draw. But the problem is that the resolution is only enhanced along the scanning direction. And from this optical design, we are not able to get the isotropic resolution enhancement in plane. So we are thinking to use deep learning to solve the problem. And the idea is that if we have a confocal input, and we can train this confocal input with this 1D sim results, right? And once we build this 1D sim model, then we rotate this input at different an arbitrary angle and apply this 1D sim model to these rotated images. And we found that the resolution enhancement always occurred along one dimension. And then when we rotate the image back, then the resolution enhancement occurred at different orientations. And then we join deconvert these different orientations, like here we use 6 rotations, then we can get 2-dimensional super resolved images rather than just 1D sim results. And here shows some examples of microtubules. So this is raw data, and here is the frequency domain, and here's the zoom in regions. And after 5 phases reconstructions, we get super resolved along this Y dimension. And with deep learning, we can predict these dimensions. And when you rotate the images, the diffraction limit input, and then apply the model and then rotate back, we found out the resolution enhancement occurs at other directions. And then we join deconvert these predictions and create the 2-dimensional sim. And when you compare these images, you see these filaments are well better resolved than just a single directional sim. And this shows the line profiles. These 2 filaments can better separate with 2D sim, otherwise it's blurred with 1D sim or the raw data. So we apply this trick to all 3 views, and now then we get triple view 2D sim. So here is a live examples with the I-10, and you can see that the triple view 2D sim can greatly improve the resolution relative compared to the raw, and we find out there's a 10-fold improvement. And here the training and testing is based on RCAM. And we applied the same method, the same deep learning trick to thicker samples on worm adults. And here is about the dense nerve regions in the membranes region. And here compares the raw triple view diffraction limit, just 1 view 2D sim and triple view 2D sim. You can see that the 2D sim, triple view 2D sim gets us the best structures and sharpest details than just even single view or diffraction limit. And so these 2 combination of the hardware and the deep learning results enable us to see these very, very details, and which is otherwise impossible with the existing microscope. So the second example is a structured illumination microscope, and that's a 3D SIM and the 4-beam SIM. So here shows the excitation pattern of wide field, and then if we introduce 3 beams from the back focal plane of objective, then we can create this structured illumination pattern not only in lateral dimension but also in axial dimensions. So here in this case, you can double the resolution in X, Y, and Z. And if you further use a mirror to reflect the beam to create interference of 4 beams, then we introduce more higher modulation frequency, and then we can further improve the resolution. And here compares the resolution measured on 100 nanometer bits and shows the intensity profile along the Z dimensions. You can see that 3D SIM greatly improve this range relative to the wide field, and 4D SIM can further improve the resolution. And we show that with 3D SIM, we can get 120 nanometer in lateral dimension and 300 nanometer in axial dimension. But the 4-beam SIM allow us to get the same lateral resolution and boost the axial resolution to less than 550 nanometer. And this shows that 4-beam SIM enables near isotropic resolution in biological samples. This example is bacteria. Compare the wide field 3D SIM and 4-beam SIM. You can appreciate that these fine structures only can be seen with this 4-beam SIM. Otherwise, it's blurry with 3D SIM and impossible with wide field. And these line profiles further confirm we can resolve these fine structures. And this example is outer membranes of the mitochondria, and the color indicates different depths. Again, we compare the 3D SIM and 4B SIM at different lateral positions and axial positions, and you see that it's greatly improved in the axial view. But the trouble again is that to get this nearly isotropic resolution, we need to use a mirror, and it's very hard to implement for live imaging and also cause actual photo bleaching and photo damage. So we seek to use the deep learning method to improve the axial resolution. And the trick is similar to the multi-view confocal microscope. The idea is to blur the lateral dimensions so that the resolution in lateral dimension is similar or close to the axial dimensions. Then we downsample and upsample this sample to mimic the real Z acquisition. And then we train the model from this data to this 3D SIM ground truth. And then once we have this model, then we rotate the input at different angles, at any arbitrary angle, apply these models, then we can improve the resolution like this, always increase along these X dimensions, and then we rotate back, then this resolution occurs at different orientations. And then when we run the frequency domain, you can see that the resolution occurs at different orientations. We take the maximum value at each spatial frequency and then inverse the Fourier transform, and eventually we can get we can recover the isotropic resolution in 3D SIM. And here shows some comparisons. Is mitochondria. You can see the 3D SIM and 4B SIM and the deep learning methods based on the 3D SIM. You can see that these better resolve these fine structures than 4-beam SIM. And here's another dual color examples based on fibroblast and Cardia 1 and Cardia 1. And you see that 3D SIM results, you cannot see these details, but deep learning predictions can see these hollow cores and different kinds of structures. And this is very close to the EM results. And again, for 3-dimensional SIM imaging, we need to combine multiple input with UNAC reconstruction to get high quality results. But for live imaging, you cannot get a good with high SNR and to image longer. So we use denoising step methods. And again, we develop multiple denoising steps. The idea is to denoise each raw images to get the first denoising model. And then based on these denoised images, we went on to construct the 3D sim. And with the high SNR, we directly construct this as a ground truth and then train a second model. And this method is better than if we just use the 15 raw SNR input as an input and this high SNR 3D construction as an output. So here we compare these different methods from low SNR, high SNR ground truth, and two-step Arcan and a one-step Arcan from 15 input. You can see that only these two-step Arcan can provide the best resolution than this one step with Arcan or this one step with density connect. So if you combine this denoising step and isotropic model, we are able to push the limit in three-dimensional super resolution images. And this example is live cells with LAM1 and lysotracker, and you can see that the low SNR 3D construction suffers many artifacts, but this two-step denoising and isotropic can follow these dynamics. So in summary, we have shown that ILN has interpretability and is similar to ILD and intermediate output. And this ILN provides better, faster deconvolution capability than those purely data-driven neural networks. But multiple step deep learning is required to deal with those low SNR images. And ILM provides 4 to 6-fold speed up over the previous processing pipeline for the joint deconvolution of large volumes acquired with dual light sheet microscope. And this method can also completely bypass artifacts induced by poor registration between views. And last, we show the multi-step deep learning approaches using KL, Arcan, DDN, and RLN can improve volumetric resolution in different fluorescence microscope, including our recent developed multi-view confocal super resolution microscope and the 3D structured illumination microscope. Finally, I would like to thank our lab chief, Dr. Harris Rolf, and other lab members who contribute to this project, Pluto and Xuesong, and our collaborators inside and outside NIH who contribute the ILN, Yue Li, and Dr. Liu in Zhejiang University in China. And thank you for your attention.
Thank you so much, Yicong. That was quite a tour de force. As always, very, very impressive to see your presentation and to see the latest from the lab. I do have a few questions. Let me get my notes back in front of me. Where did they go?
You see?
There it is. Okay.
Can you hear me? Mm-hmm. Sure.
We can hear you perfectly. Yes, perfect.
Nice. Mm-hmm.
So this is absolutely breathtaking, and it seems like every couple of slides we are presented with another amazing kind of step forward in compressing that triangle that you presented at the beginning. So you're systematically figuring out ways to make it possible to do high resolution, temporal and spatial resolution, and do it quickly without damaging the sample. So it's impressive and really a pleasure to watch. I do have one about accessibility, because this is, it seems like a very custom-made piece of hardware and a very custom-made piece of software. So what are you doing to make this more accessible to the broader community? Because it seems like such a powerful set of tools and developments that you've created now.
So this split to a few things. For example, the dice beam actually has been commercialized and people can access this system. And even there are many so-called multi-view light sheet microscope, and I think they can use they can train if they can acquire data run whatever the ground truth. And the other thing is, as I mentioned in ILN, it's quite generalization. So you don't need to be very specific to the structure you are imaging. you can create a synthetic phantom and just blur with your system PSF. So that's another way you can do. So regarding our recent paper about the multi-view light sheet microscope and structured illumination microscope, yes, I do agree that hardware is quite hard to copy too, but we also try something funny, something interesting thing. So we apply the model trained with on our microscope on our confocal, and then apply to those commercial spinning discs, and then the model works. It also improve the lighter resolution, improve the ICO resolution. So it's amazing at this moment. And for the last part about the 3D structural illumination, the key point is we can improve the ICO dimension. And I think that's is powerful. We haven't tested for other modality, but we believe this same trick can be used for any microscope if they want to, because everything then is from the lighter view to improve the ICO view. So I believe it's not limited to our microscope. It's just the method. If you can apply, if you can get a training data set by your image, and feel that you don't need a bunch of training data set, maybe 10 or 20 volumes is sufficient to recover the resolution. Yeah. And we of course we hope if Leica can commercialize our system. Right? So
yeah. Thank you for the hint. So one of the, as you say, tricks that you're using is stacking of deep learning models, and you do kind of one step for denoising, another step for the joint deconvolution, and sometimes a third step. But can you give some intuition? Why is that better than just doing end-to-end in one go? So you might sometimes not have the ground truth. I appreciate that, the endpoint ground truth, but sometimes you have it. So when you do have the endpoint ground truth and the input data, why not train a model to go all the way to the end instead of multiple models to do each step?
Yeah, I think this is a good question. We're also puzzled by this, but we think because a single neural network can, because there's need to optimize the kernels, right? And some kernel is only for the denoising, some kernel is only for deblurring, deconvolution. So if you train them together, then kernel will just need to be compromised, so you cannot get the best results. But if for this, even for just Arcan, you just 2 steps, 3 steps, and each network just optimize or learn its own based kernels at different layers, then maybe combine this cassette or sequential, maybe give you the best results. So that's all we feel.
Okay.
Unless maybe you use very deep networks, maybe you can learn more, right? You can just one step, but
right, indeed. I mean, maybe it wasn't clear to everybody, but these are not particularly deep, and you're not training with a tremendous amount of data. So this is really kind of usable by the general user of microscopy. It's not a Google-sized type of project. It's a reasonably easy to get amount of training data, and you can run these algorithms on a good computer locally. You don't need huge cloud resources.
Yes, I agree with your point. Yes, so you split to multiple different small tasks. That might be easy.
So one last thing, looking into the future. You've made, together with the team, tremendous progress in the last 5 years in this AI microscopy world. What kind of experiments would you say are now possible that weren't possible 5 years ago? First part. And the second question is, what do you expect will be possible in 5 years that right now is not?
Yeah, I have to say, even 5 years ago, I'm a microscopist. I'm not any deep learning computation guy. But 5 years ago, I attended a deep learning course, and then because at that time it's hot, and then we think, okay, how to use the deep learning to improve our imaging performance, right? So at that time, we start to think, and then we are very lucky to have a collaboration with you, and then develop these so many promising tools. And so 5 years ago, we definitely cannot imagine we have these, especially when we do the when we try to implement the multi-view confocal microscope. We haven't thought about the deep learning, but then once we have data, we and then we seek the use of deep learning, and then we achieve those results. And for the next 5 years, good questions. We never know, and then let's see.
Okay, all right. Very good. Thank you so much. Yicong, pleasure to have you on the AI Microscopy Symposium. Thank you so much for your time and all your insight now in the Q&A. I will thank you one last time, and we'll progress to the next speaker. Our next speaker is Parmida, and she comes, this is Dr. Parmida Ghahremani. Sorry for my pronunciation. She's a senior data scientist at the Memorial Sloan Kettering Cancer Center in New York. And she did get her PhD from Stony Brook University, and also has a master's in computer science as well as a PhD in computer science. So absolutely expert. We'll be hearing about some of the developments that she's been involved with, and she's been involved in quite a few exciting ones, including Neuron Construct, which we'll post in the chat in a moment so you can check it out, and DeepLIF, which is part of the story today. So without further ado, I pass it over to Parmida, and I will be back at the end for the Q&A, final Q&A session of today. Thank you.
Hello, everyone. Thanks for having me today. My name is Parmida Ghahremani, and I'm a senior data scientist at Memorial Sloan Kettering Cancer Center. Today, I want to talk about DeepLIF, a multitask deep learning model that infers more informative image representations and at the same time segments and classifies the cells for more accurate IHC quantification. Interpreting images of tissues and cells at a resolution higher than the naked human eye is the core of pathology and essential to diagnose diseases. Due to the rapid progress of medical imaging techniques over the past decade, pathology has been improved considerably by creating digital slide scanners that can capture whole slide images. Pathology plays a vital role in cancer diagnosis and treatment. The pathological analysis is performed based on the visual interpretation of the tissue sample under the microscope. Due to the difference in visual perception and clinical training, pathologists can have conflicting opinions about the cancer diagnosis and treatment. Over the past decades, computer vision and deep learning approaches have been developed to assist pathologists in assessing tissue sections. The computational pathology has several challenges. The first challenge is the large size of the whole slide image. For instance, a single image acquired at 20x magnification can contain billions of pixels, while the area of interest can be as small as a few thousand pixels. The next challenge is the lack of a universal image format. Currently, there are various whole slide image scanners from different vendors for both bright field and fluorescence imaging that capture images using different compression types, sizes, illuminations, objectives, and resolutions, and output them in different file formats. The next challenge is the high morphological and color variance and having artifacts. And the last challenge is the large amount of time and expense required to obtain expert-derived labeled images. Computer vision has been used for many tasks, ranging from low levels such as edge detection to high levels such as complete scene analysis. In the last decade, there has been lots of improvements in artificial intelligence. Because of major progress in deep learning, tons of advancements in GPUs, and release of large labeled datasets. The application of deep learning in medical image analysis involves various tasks, including object detection, segmentation, classification, and registration. Immunohistochemistry, called IHC, is used in histology to detect the presence of specific protein markers that can assist with accurate tumor classification and diagnosis. Where H&E and special stains are non-specific, IHC is directed to a specific protein marker. Pathologists look for the presence or absence of particular markers. Multiplex immunofluorescence, called MPIF, is used for simultaneous detection of several target proteins in the same cell. This generates complex spatial information of a tumor or tissue sample, and it can be used to visualize the spatial distribution of proteins within a single cell. Typical workflow of IHC quantification in commercial tools such as Halo consists of several steps. First, IHC depth deconvolution needs to be performed on the input IHC image. Then, nuclei are segmented in the hematoxylin channel. Next, a threshold setting is found for the brown nub stain to separate cells from the background noises. And finally, the cells are classified based on the threshold found in the last step. Here, we present DeepLife, a simpler workflow for IHC quantification. Given an IHC input image, DeepLife generates 4 modalities and the classified segmented mask. DeepLife performs IHC quantification in just one step, and it does not require error-prone IHC deconvolution or manual thresholding steps. DeepLife is a multitask deep learning technique that uses co-registered IHC and MPIF data to simultaneously translate low-cost IHC images to more informative MPIF representations and accurately segment the cells and classify them for more accurate and reproducible IHC quantification. Given an IHC input image, our multitask deep learning framework simultaneously infers the corresponding hematoxylin channel MPIF DAPI image that has only the cells inside it without any background noises. MPIF Lab2 image, which is the new marker and shows the boundaries around the cells. And then MPIF marker image. And finally, the classified segmentation mask, where the red cells show positive protein expression and the blue cells show negative cells. We first had to align the IHC images and MPIF data to create our training set. So we designed a registration framework and co-registered 350 IHC pairs of bladder and 350 pairs of lung data. Our training set contains pairs of IHC, hematoxylin, Dept, Lab2, marker, and segmentation mask. Deep Life consists of several components. The main component of Deep Life is GANs, which are generative adversarial networks. GANs are used for generating synthetic data. It consists of a generator that captures the data distribution and a discriminator that estimates the probability that a sample is real or fake. We use residual networks for the translation task. It consists of residual blocks, and it has skip connections, skipping the training from a few layers and connect directly to the output. In the segmentation task, we use unit structure, which has shown great success in segmenting biomedical images. It's a fully convolutional network that has a contracting path to capture the context and a symmetric expanding path that enables precise localization. Deep Life consists of two main parts: translation task, which is used for inferring the modalities. As you can see here, given an IHC input image, we have 4 generators that infer hematoxylin channel and MPIF data, including Dept, Lab2, and the marker image. The generators in the segmentation, in the translation task, are residual networks. Then we have the segmentation or classification task, which is used for generating the classified segmentation mask. Here we have 5 different generators that create a classified segmentation mask from the input IHC image and the inferred modalities in the translation mask, and then using a weighted average module creates the final classified segmentation mask. The generators in the segmentation task have unit architecture. For each generator in the translation task and in the segmentation task, we have a corresponding discriminator. To address high frequencies in the image, we use a patch-GAN discriminator that only penalizes structure at the scale of patches, meaning that it classifies patches in an image as real or fake. So we run this fully convolutional discriminator across the image, averaging all responses to provide the final output of the discriminator. To optimize our network, we alternate between one gradient descent step on discriminators and one step on generators. In all defined tasks, including translation, classification, and segmentation, the network generates different representations for the same cells in the input image, meaning that all tasks have the same endpoint. Therefore, we use a single optimizer for all generators and a single optimizer for all discriminators. Using this approach, optimizing the parameters of the task, the more clear representation of cells improves the accuracy of other tasks, since all of them are optimized together at the same time. In the translation task, we have 4 generators to infer hematoxylin channel, Dept image, Lab2, and the marker image from the given input IHC image. We have a discriminator corresponding to each generator, which is responsible for discriminating the generated images by that generator. The objective of the conditional GAN in the image translation task are defined as you can see here. We use smooth L1 loss to compute the error between the predicted value and the true value, since it's less sensitive to outlier. And then we calculate the objective loss function of the translation task based on the conditional GAN loss and the L1 loss for all generators and discriminators. In the segmentation or classification task, we have 5 generators producing 5 individual segmentation masks from the original IHC image, the inferred hematoxylin image, inferred MPI of Dept, inferred Lab2, and inferred marker image. Then we calculate the final segmentation mask by calculating a weighted average of the segmentation mask generated from these 5 individual segmentation masks. Again, we have 1 discriminator corresponding to each generator in this task. Here, we use LSGAN loss function as it solves the problem of vanishing gradients for the segmented pixels on the correct side of the decision boundary. We again use smooth L1 loss, and so the objective loss function of the segmentation classification task is calculated using the L1 loss of the final segmentation mask that's calculated here, and the LSGAN loss function of the final segmentation mask and the discriminators. The final objective of the diploid is calculated using the loss of the translation task and the objective loss function of the segmentation task. We tested diploid on different testing datasets. The first one is the BC dataset, which contains case 67 stained sections of breast carcinoma with positive and negative cell centroid annotations. So we randomly selected 41 images of size 640 by 640 and segmented all these images. Then we split these tiles into 164 images of size 512 by 512. We also tested diploid on nucleic dataset with a publicly available CD3, CD8 IHC dataset. We used the nucleic training set containing 671 IHC patches of size 256 by 256 that all are extracted from the LYON19 dataset. To evaluate our model against state-of-the-art methods, we define these metrics. The first one is the pixel accuracy, which is calculated from the number of the true positives and false positives and false negatives. We also calculated DICE score, which is giving more weight to the true positives. We then calculated the intersection over the union metric, but we calculated it for each class and calculated the weighted, the average of these IOU for these classes. We also calculated aggregated Jaccard index with an object level metric, meaning that we calculated the IOU for each cell separately. Then, because actually the goal of deep life is IHC quantification, we calculated the IHC quantification error, which is the difference between the IHC quantification percentage of the predicted mask and the real mask. Testing all these methods on the BC dataset, you can see that deep life outperforms the state-of-the-art models, including NNUNet, recently published in Nature, mask CNN that has shown great performance on semantic segmentation tasks, UNet++, LinkNet, and FVN, and also if it has deep life has less error in IHC quantification compared to the rest of the models. Here, you can see two sample images taken from the BC dataset. The predicted classes are shown in different colors, where blue represents Ki67 negative and red represents Ki67 positive cells. And the hue is set using the log 2 of the ratio between the predicted area and the ground truth area. Cells with two large areas are shown in dark colors, and cells with two small areas are shown in a light color. For example, if the model correctly classifies a cell as Ki67 positive, but the predicted cell area is too large, the cell is colored in dark red. And if there's no cell in the ground truth mask corresponding to a predicted cell, the predicted cell is shown in yellow, which means that the cell is misclassified. It means that the cell segmented correctly but classified wrongly, or missegmented, meaning that no cells in the segmented cell area is available. Here you can see two example images taken from the BC dataset and the inferred modalities generated by the deep life. And here you can see the results generated by deep life and the state-of-the-art models. And as you can see, deep life outperforms the state-of-the-art models in terms of the AGI and pixel accuracy in these two examples. And you can see it also that the segmentation and the classification is more accurate when we use deep life. We tested deep life on IHC images of various markers captured in different clinical settings. We visualized the structure of the testing dataset by applying t-distributed stochastic neighbor embedding to the image styles tested on the deep life. To do that, we first extracted the features from each image using the VGG16 model, and then we applied PCA to reduce the number of dimensions in the feature vectors. Next, we visualized the image data points based on the extracted feature vectors using TSNE. As you can see here, the training dataset has completely different characteristics compared to the testing dataset. And yet, deep life is able to adapt to images with various resolutions, color, and intensity distributions, and magnifications captured in different clinical settings, and successfully segment and classify the heterogeneous collection of the testing sets covering 8 different markers. And you can see here the classified segmentation mask. And as shown, DeepLife can accurately segment and classify all these images, which has different staining and are taken from different markers. We also tested DeepLife on microscopic snapshots from a digital microscope that was stained with K67 and PD-L1 markers. On the right side, you can see one sample image of the PD-L1 marker, and on the left side, you can see one sample image of the K67 marker. As you can see here, DeepLife is able to infer the modalities and accurately segment and classify these macroscopic snapshots. We also tested DeepLife on LYON19 challenge dataset that contains 441 IHC CD3, CD8 breast, colon, and prostate region of interest with various staining and tissue artifacts, and they were collected from 8 different institutions. Unfortunately, they have not provided the annotations. As you can see here, DeepLife again can infer the modalities and segment and classify the images of the CD3, CD8 marker with artifacts in them and different staining and taken from different tissues, while it's only trained on Ki67 marker images. We also tested DeepLife on PathoNet dataset, which contains IHC Ki67 breast cancer images. And it contains manual centroid annotations, which are created from the consensus of multiple pathologists. These images were acquired in low resource setting with microscope camera, and as you can see, DeepLife is able to segment and classify the cells with more than 95% accuracy. We also tested DeepLife on Human Protein Atlas IHC Ki67 dataset. This dataset covers 6 different cancers from patients with different genders and ages, and it actually covers different type of staining and intensity, and with different IHC quantification score. As you can see here, DeepLife can give you an exact IHC quantification score like this, which is 49%, where pathologists only provided a range for the IHC quantification score, which is 25 to 75%. So here with DeepLife, we can have more accurate IHC quantification. We also tested DeepLife on H&E images, and surprisingly, DeepLife can segment the H&E images while it's trained on IHC images. And it's because the deploy first infers the modalities, including the hematoxylin, Dept, and Lab2 image that would help in segmenting the pathological images. After training the first version of deploy, we figured out that it has issue in separating positive cells in cluster of high contrast positive cells, as our training data lacks this kind of information. So we designed our IHC generator model. It gets 3 images, hematoxylin, Dept, and the classified segmentation mask as the input, and generates the synthesized IHC image and its corresponding marker image as the output. After training our model, we generated synthesized IHC images with cluster of positive cells by converting the negative cells, which are shown in blue, to positive cells, which are shown in red. And then using this IHC generator model that we already trained, synthesize IHC images and their corresponding markers. As you can see here, this is the original image and its segmentation mask. So we randomly selected some cells from the segmentation mask and converted them to positive cells. And then we used this model and generated these synthesized IHC images and their corresponding markers. We also synthesized the staining and intensity of IHC images by changing the contrast and intensity of the hematoxylin image, and then using this model to create the synthesized IHC images. There are different ways to use deploy. The first one is our GitHub repository. You can access the code and the instructions how to use this code and to run your data using the pre-trained model, or if you want to use the deploy to train it on your dataset. We also have a website that you can easily upload your images, and it gives you the inferred modalities and the classified segmentation mask, as well as the IHC quantification score and number of the positive and negative cells. We also have an ImageJ plugin that gives you more options and more features. For example, as you can see here, you can select a region of interest to do the segmentation in that region. And then you can change some thresholds like the cell size or the probability of the cell to be a cell exactly or not. And so using our plugin, you can get the best version of the segmentation mask, the one that fits your needs. This concludes my talk. Thank you for joining today. I look forward to answering any of your questions in the Q&A session. Joining today. I look forward.
Hello. Hey, welcome back. Thank you so much for the presentation. That was quite special. It was one of the first ones we had focused on this topic of multiplexing and virtual staining. Parmida, congratulations on the work that you're doing together with your group. I have a few questions that some of them have come in, others I have myself. I'm curious about, let me get my notes here back. Okay. One of the things I'm curious about is so you've shown how to do this for nuclei. It looks like it's mostly nuclei detection, segmentation, and you can predict also the fluorescent markers. This is already a huge step forward. I'm curious if you have tested this versus other methods, like you mentioned Halo at the beginning, other standard, not machine learning methods, and also other machine learning methods like CellPose or StarDist, which use different neural networks, but are pretty popular in the life science space, maybe not so much in the digital pathology space. So I'm curious about those tests. I saw the ones you have on the paper and the ones you presented today, but those are specific networks, not so much a full pipeline. Like CellPose or StarDist, they have really tried to optimize the whole stack to make good segmentation. So that was my first question.
First, the question about the Halo. Yeah, we tested Halo. We tested some other frameworks as well. And actually, we found out that this deep life outperforms those segmentation algorithms as well. And about the CellPose. CellPose and DeepCell are designed to segment the cells in Dept images and MPIF images, not IHC images. So Deep Life somehow is the first one that is trying to segment the IHC images, which is much harder. Actually, one of the things that we tested is that to collect the segmentations for the generating the ground truth dataset for Deep Life, we actually tried those DeepCell and CellPose and Halo on the Dept images, because you cannot generate some preliminary segmentations with IHC images. There are not any available model that can do that very well. Yeah, and actually Dept segmentation at Dept channel is much easier than the IHC image. And yeah, it's impossible to compare our model with the DeepCell and CellPose because we are doing different things.
I see. Okay, sorry, there, I was muted there. I appreciate that. What about the Allen Cell Institute? So they do go from, it's not exactly the same thing because they're looking at transmitted light instead of immunohistochemistry, but there they also transpose. So that's also a different approach. So it wouldn't make sense to compare there. Never mind. So very interesting. Another question or another topic related to what you showed, and I think you kind of touched that on the paper, is what is the feasibility of going towards the detection also of different organelles and other types of labeling, not just the nuclei or the membrane, which have a pretty clear representation when you look at immunohistochemistry, but not so clear if you're doing, let's say, mitochondria or some other, a vesicle of some sort, or ER or any other organelle. What about those? Do you think those will be possible to predict also from immunohistochemistry to get the pseudo fluorescence and then do segmentation of those, or that's beyond the technology currently?
No, exactly. That's what we are right now trying to use Deep Life for. We are actually we have the Deep Life in its version that you can see in the main GitHub repository, only doing some specific things like generating the hematoxylin, the Dept marker, and Lab2 and the classified segmentation mask. But we also have a configurable branch in our GitHub repository that is actually the goal of this branch, that we tested on different data sets, different type of works, because we don't want to generate only these 4 type of images. What we are actually right now are doing is testing it on another data set and just trying to infer the immune marker and tumor marker from the hematoxylin channel only. And that's another thing that we are testing with Deep Life. And in the configurable version, you can just say what's the number of the modalities that you want to get, and if you want to do the segmentation or not, or do you want to do the classification. So it can be used in different projects, and yeah, it can definitely do that, I believe.
Okay, and one last question before we close off. I'm about multiple labeling. When you have a high-plex image with, let's say, 20 channels, and 3 or 4 of those represent a particular region of interest, have you tried to create a segmentation that goes directly to that using this approach, using your platform? So where 3 or 4 channels are representing a positive region, not a positive cell, but a positive region. Does that work as well with this approach?
That's exactly why we have the weighted average module. And so with the weighted average, you can just set the value of those channels as 0 in the weight of the generating the final segmentation mask, and then they will not do anything in generating the final segmentation mask. And that's what we do in some cases.
Very exciting. It's clear that now the digital pathology or spatial biology environment community is very, very active using machine learning to try and get to better performance than before. Like I was asking Yicong in the previous presentation, I'm wondering where you think this is going. What is the future in this space? What do you think will be possible in a few years' time that isn't currently possible?
Honestly, by now there are a lot of works that has been done. For example, people are doing cancer treatment using the results that the deep learning models are actually generating. And so I think and it's been maybe less than 10 years that deep learning has been introduced and used for these I mean, even for the healthcare, I think it's maybe less than 5 years that deep learning has been used. And so I believe that in future, in maybe less than 5 years, we will see a large improvement in the diagnosis and treatment part of the healthcare.
Very well. Well, I'm looking forward to welcoming you back at some time in the future to see the evolution of your platform and the applications, the different applications and models that you built. Thank you again for your amazing.
Thank you for having me.
A pleasure. It's our pleasure. So with that, I wanted to thank all the speakers, including our Melbourne, Australia-based speakers, Vijay and Afshin, who were the first presenters today. Then we had Yicong presenting from the NIH in Bethesda on the east coast of the US. And then now we finished up with Parmida, also on the east coast in New York City. I myself, I'm in Lisbon right now, and the last thing I wanted to mention is that we will be having a future AI Microscopy Symposium. Will be the 8th edition. We'll be planning that for later in the year, but very soon we'll have the 5th MeCam live stream, which will be with me. So I'll be talking about image analysis. It will be a live session. The aim there is to look at images that are pretty complex. Then we do image analysis there live, and also showcase some of the latest systems that we have developed here at Leica. Specifically here, we're going to be talking about Mika, which is the micro hub. Does both confocal imaging as well as wide field with deconvolution. So it's a wonderful new system that we just introduced earlier this year. And this particular episode will be about image analysis of images that come off that microscope. So with that, I want to thank everyone that participated, attended, and helped organize today's session. And I pass it over to the lab groups team that has done a wonderful job to host us today. Thank you.
Thank you, Luciano. And thank you again to all of our speakers for their time today and important research. We would also like to thank LabRoots and our sponsor, Leica Microsystems, for underwriting today's educational webcast. Before we go, I'd also like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speakers via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye.